Hey everyone, welcome to my PhD defense. If everything goes well, this is going to be released as I'm live presenting my thesis to my committee as well as some members of the public. However, there are some parts of my talk that are unpublished, and so I wanted to create a separate recording with a incredibly similar presentation, but just excluding those sort of more secret details. So this is my PhD defense presentation, excluding some details. And without further ado, let's get started. I'm calling this presentation Smart Yeast, and I'll explain exactly why it's called Smart Yeast within the talk. But my sort of academic name is Nicholas R. Robertson. If you ever want to look into my Google Scholar, I'm a PhD candidate in the Bioengineering department at the University of California, Riverside. I wrote about a 200-page thesis. I don't know how quickly that will be open to the public, but the outline of my thesis is, of course, an introduction and a conclusion chapter. Typically, theses have about 5 or 6 chapters. So my first real chapter is a review on functional genomic screens, focusing specifically on CRISPR screens and yeast. My second chapter is a previously published work that's a functional genomic screen in the yeast Yarrowia lipolytica. My fourth chapter focuses on some work that I did towards the beginning of my PhD on designing biosensors for metabolic engineering targets. And then finally, the fifth chapter combines chapter 3 and chapter 4 to use these biosensors for functional genomic screening. So I want to focus mainly on chapter 3 and chapter 4. One, again, is a previously published work. I'll make a full video on that very soon if you want to see details, or you can go find that paper as well. And then chapter 5, which will soon be published, which is sort of the culmination of my whole thesis. And this is really exciting because I've been working on something along these lines since the beginning of my PhD. So usually when you start out in research, there's jokes about this everywhere, but usually when you start out in research, you have some idea, and by the time you get to the end of your project, it's much different than what you started with. But in this case, I think it's stayed pretty close to the same throughout my whole PhD, which feels really cool. So I want to share with you why I love yeast and why you should love yeast too. So we often refer to microorganisms as microbial cell factories, or yeast can be referred to as yeast cell factories, because that's exactly what they are. They're microscopic factories that can convert sort of lower value products into higher value products. Normally, we feed yeast things like glucose. So in the US, we grow corn and we process it to get the glucose, and then we can feed that to yeast to make things like ethanol. So that's a very traditional pipeline. We've been doing that for, decades, or really we've been doing that for thousands of years in the form of beer and wine. But yeast can also consume waste substrates from other industries. So we'll focus in this presentation a bit on acetate, which can be a waste product from some industries, but as well as hydrocarbons. So fatty acids and various other hydrocarbons can be consumed by some yeast to make higher value products. And if you aren't focused on making a higher value product, you can use yeast as well as other fungi for bioremediation purposes. So you might have, like an oil spill or something like that, or pesticides in a given area. And maybe there's some yeast, either modified or unmodified, that can break down those compounds and convert them to less harmful products. And then on the production side of these yeast cell factories, we're able to produce things like biofuels. So if you go to the gas station and you buy gas, it's usually 10% ethanol. And that ethanol, 100%, comes from yeast. So ethanol is made from yeast. And there's a lot of work as well with making biodiesels from yeast, as well as biojet fuels from yeast, which is really cool. Yeast can also make fragrances and flavorings and food molecules as well. So you can make things like whey protein. So there's a few startups that say that instead of growing an entire cow to make your whey protein, you can just genetically modify yeast to make that same protein in a much more environmentally friendly and potentially a much more affordable way. I'll focus today also on terpenes. So terpenes are commonly found in cannabis products, and they're also in hops. So in beer, they're very prevalent in beer. But in a lot of foods that we consume, especially processed foods, they'll have terpenes added to enhance those fragrances. So, for example, limonene, you might guess smells like lemons, but it smells like oranges, which is cool. Linalool smells just like honeysuckle. That's my personal favorite terpene. And there's dozens of others. So like beta-myrcene smells like hops. It has that very distinct IPA or hoppy flavor. And then like geranic acid smells just like green apple flavoring. So there's a lot of really cool terpenes, and they have very specific properties. And some terpenes are also precursors to valuable chemicals. So some of these are things like FDA-approved drugs. So we'll focus a little bit on terpenes in this presentation. And then finally, you have sort of the highest value product that you can get from these yeast cell factories, and that's medicines. So all insulin, as far as I know, is made in yeast. Genetically modified yeast make insulin. It used to be that we had to grind up a pig pancreas to get insulin. And then eventually we started making insulin in recombinant mammalian cells, so we can make it in CHO cells, so Chinese hamster ovary cells. But now we can make it pretty easily in yeast, which is much less likely to be contaminated, can make more of the protein. We can also make new drugs like Ozempic in yeast. So that new weight loss drug Ozempic, that's made in yeast. So very cool stuff. So now I want to introduce you some of the specific characters, some of the specific yeast that I work with. So the first is Saccharomyces cerevisiae. This, humans have worked with this yeast for thousands of years as it's used to make bread and beer and wine, right? In academia, it's been heavily utilized because of some of the molecular machinery that this specific yeast has, as well as it just being a very well domesticated strain. So baker's yeast, brewer's yeast, Saccharomyces cerevisiae, whatever you want to call it, it is the conventional yeast. So it's the model organism for the group of yeast. Next, we have Cluveromyces marxianus, which is my personal favorite. I think it smells fantastic. It smells like a Hefeweizen if you've ever had that style of beer before. It was originally isolated from the dairy industry, and it can grow about twice as fast as Saccharomyces. So that means you make your product about twice as fast, and all of the experiments you do happen about twice as fast, which is great. And it can also grow at really high temperatures, up to around 50, maybe even 55 degrees Celsius, which is hot. Right? So not many things can survive at that temperature. So for industrial applications, this is really great for many reasons, but one of them is less likely contamination when your organism grows at these high temperatures. And then finally, we have Yarrowia lipolytica. It's a stinkier yeast that was originally isolated from a sewer. When it grows on petri dishes, it has pseudo-hyphae, or it has a hyphal state. It can grow in a hyphal state. It's called a dimorphic yeast, so it can grow in a single-celled state like baker's yeast, and it can also grow in a hyphal state like filamentous fungi. And so it can switch between those two modes. So you can see on a petri dish, it gets these sort of wrinkled morphologies on a plate, which is cool. For industrial applications, it's very useful because it can accumulate up to around 90% lipids in its cells. So it's an obese yeast. We call it an oleaginous yeast. So because of this propensity to produce these lipids, it also likely has a very high acetyl-CoA pool, and acetyl-CoA is a metabolic precursor to a lot of other valuable chemicals. So this sets the stage. These are all of the yeasts that we're going to work with in this presentation, and all of the yeasts that I've personally worked with as well. Next, I want to define exactly what I mean by smart yeast, because this is not technically something that's seen, a term that's used in the literature, but it's a term that I like to use and that I'm using for this presentation. So commonly, we want to produce some product in yeast. Sometimes we get lucky and it's a colorful compound. So perhaps it's something like beta-carotene, which has a deep red color. And so we can see when yeast are making that product because the yeast will be red, right? Let's say we make a strain of yeast that makes some beta-carotene, so the yeast is a little bit orange, and then we do some mutations to a group of that yeast making our beta-carotene, and we expect some of them to stop making beta-carotene because of some particular mutation, and we expect some of them to make more beta-carotene because of some different mutation, right? So then we can take those cells and put them on a petri dish and look for really, really deep red colonies. And that strain should be making a lot more beta carotene than the other colonies on that plate. So we can look at this plate here, this colorful petri dish, and say, okay, this colony is very deep red. This one probably makes the most beta carotene, right? This is best case scenario currently. But more often than not, we're producing some compound that isn't colorful. And so you can't just look at this petri dish and know which colony has a mutation that produces more of your product. And so what you end up having to do is pick a single colony, grow it up, and then send it through some very expensive analytical equipment on the order of hundreds of thousands of dollars, and then figure out how much product your yeast is making. So the problem that exists here is that typically with most products that we make, we can screen through maybe 100 mutants per month with this manual analytical chemistry method. If we're lucky, we have some compound that has some, phenotype that's very visible, like, a red color. And so in that best case scenario, we're looking at hundreds of thousands of mutants per month. So what I'm suggesting here is a yeast that has some way to tell us or report how much compound they're making. So we do that by adding a biosensor. So a biosensor is going to be activated by the molecule that we're trying to produce. And if a yeast cell, like in the teal here, if a yeast cell is producing just a little bit of that compound, then we get almost no activation of the biosensor and the yeast cell is quiet. But if we make a lot of that compound, then that biosensor is very active and the yeast screams to us, pick me, pick me, pick me. And so biosensors allow yeast to tell us, or again, report how much product they're making. And this allows us to screen through on the order of trillions of mutants per month. So, several orders of magnitude more mutational screening capability than the traditional methods. So this is what I mean by smart yeast. So in this presentation, we'll be building a smart yeast platform. And we'll be doing that with this class of biosensors called Peer 1 biosensors. And I'll have a couple of videos coming out soon, one more general video and one more specific video about how these biosensors work specifically. But basically you can imagine Peer 1, well, first Peer 1 is originally a plant hormone receptor that detects a chemical called abscisic acid, or ABA. And so the protein sort of looks like this, and then the chemical comes in and binds within what we call a binding pocket. And you can see that in the picture on the left. So you have, in the blue is Peer 1, and in the yellow is ABA, or abscisic acid, and it's binding in that binding pocket really snug, right? So researchers, over 15 or about 15 years ago now decided to mutate some of these amino acids within the binding pocket of this protein to change the shape of that binding pocket, to change the chemistry of that binding pocket, to detect new chemicals. This is how we make biosensors typically. This is really cool, and they found that if they mutate some of these amino acids, they actually break the biosensor. And this was an indication that they could mutate other ones to be receptive to new chemicals. And so you can see in this image on the right, this is called a beta-galactosidase stain. I have a few videos of what this means and how to do this on my channel. But basically you have machinery within the yeast. These are colonies of Saccharomyces cerevisiae. You have machinery within the yeast and a particular biosensor within the yeast that will allow the yeast to stay white if there's no activation of the biosensor, and allow the colony to turn blue if there is activation of the biosensor. So you can see this top row is Peer 1 wild type, so just a normal Peer 1. If there's no chemical added to the agar, you get a white colony, no activation of the biosensor. If you add abscisic acid, or ABA, then you get a lot of activation of that colony. However, if you do certain mutations, so this is sort of a mutation notation, this superscript. So P is the original amino acid at the 88 position, and it's replaced with an S amino acid. I don't remember what P and S is. I think it's phenylalanine and serine maybe. Anyway, at that position, there is a mutation that's within the binding pocket. It's at this 88 position, which is between this 89 and 87 position. So somewhere around here, they're mutating this amino acid, and it no longer binds abscisic acid. So they've broken that binding pocket of the protein. Okay, so very cool. We can change, how this biosensor works. And so I've had to block out some of the unpublished data here, but the point here is that these Pier 1 biosensors are flexible. So this is one of the really big advantages of this platform. They're flexible to new ligands. So we're not looking at chemicals that are slightly different than abscisic acid. These are very, very, very different chemicals. So these Pier 1 biosensors can be sensitive to things like natural cannabinoids, so like CBDA in this case, so related to CBD. We also have THC biosensors, also synthetic cannabinoids like spice. This was a big problem in public health about a decade ago. And then organophosphates, which is a class of pesticides. And you don't have to be an organic chemist to look at these and see that these are very different molecules. And if you could see what's behind these boxes as well, you would really understand that these are very, very different molecules that different mutants of Pier 1 can detect. So things like synthetic opioids, so you can think things like fentanyl, and then particular environmental contaminants, and I can't specify what those are right now. This Pier 1 biosensor platform is flexible to a variety of ligands, which is really, really awesome. And every day we realize how flexible this biosensor is. And I have more videos, again, coming out on that that should provide a little bit more enlightenment there. So be sure to subscribe to the channel. Okay, so now moving on to the portability of this Pier 1 biosensing platform. So of course this platform was, this system was originally from plants. So Pier 1 was from plants, it was a hormone receptor, but we can mutate Pier 1, add new molecular machinery, and put it back in plants to give it a new function. So in this case, we have a mutant plant that has a biosensor for a chemical called azinphos that drives the transcription of a gene called Ruby that produces a red protein. So when you add azinphos to this plant, then the plant turns red. And so the sort of big long-term idea of this is you could have plants in a field that detects a particular environmental contaminant, and the plants sort of tell you, or they report when that contaminant is cleaned up or when that contaminant is present, right? So really cool stuff can move into plants. It can also be used in vitro in an ELISA-like assay. So in vitro meaning outside of the cell, and then ELISA-like assays are commonly used lab test methods that allow you to rapidly identify the presence or absence of a chemical. So they're commonly used for like drug tests or for metabolic tests for your blood or something. So we've shown that we can use these biosensors for ELISA-like assays to detect compounds in urine, saliva, blood. So, this is really functional as well. And then finally, we've also used this as a metabolic engineering tool. So if you're producing a product in yeast, yes, you want to produce a lot of that product, but not necessarily at the expense of the growth of the cell, right? So ideally, you want your cells to grow really quickly, and then you want to be able to flip a switch and start production of your chemical. And there are some tools to do this already, but there's some disadvantages and advantages of them. But we showed that we were able to use the Pier 1 biosensors as what we call metabolite valves. So we can turn up the growth at the beginning of the experiment, and at the end of the experiment, we can turn down the growth and turn up the production of the compound that we want. So really, really cool stuff. I have a video coming out soon on transcription factor-based biosensors. Those are very useful for certain things, but they are not nearly as flexible as what you've seen here, and not nearly as portable as what you've seen here, at least in general. And again, Pier 1 biosensors have only been a thing for maybe 15 years, so I want everybody to get excited about these. This slide is sort of explaining how we're doing the screening, so we'll talk a lot about libraries in this presentation. One of these libraries are mutant Pier 1 biosensors. So libraries are just a set of slightly different, either proteins or cells or DNA sequences or whatever. So in this case, we have a mutant Pier 1 library where that binding pocket is mutated in several different ways. We slap a bunch of terpenes. We have several petri dishes with different terpenes in them, and then we put our library of yeast that contain the mutant biosensors on those plates, and then the cells that respond to the chemical in the agar will grow and the other cells will die. So in this story, we screened 24 different terpenoids. So these are monoterpenoids that would be relevant for metabolic engineering. So we screened 24 of them, and from screening 24, we got sensors for 8 of them. So this is unpublished data, so I've had to block it out. But the point here is, not only can we find biosensors for several different kinds of chemicals, but we can find several different biosensors within a class of chemicals for slightly different chemicals. So, we might be able to detect one terpenoid with this biosensor, but it doesn't detect any of the other similar terpenoids. So that's the specificity as well. That's part of this system. Okay, so we'll come back to the Peer 1 biosensors and how they'll implement into our biosensor-driven genome-wide screen in a minute. But first we need to talk about genome-wide screens and how these work. So you can do genome-wide screens or functional genomic screens in a lot of different ways. The newest, most modern way you can do it, in my opinion, is with CRISPR-Cas9. So you've probably heard of CRISPR-Cas9 if you clicked on this video. But basically it's a way to cut DNA. And I think a lot of people get exactly what CRISPR does very confused. The only thing that CRISPR-Cas9 does is cut. That's all it does. It has a very specific targeting system to cut, but that's all it does, is it cuts a DNA sequence. So you have basically a barcode called a guide RNA that tells Cas9 where to cut. Cas9 is what's called an endonuclease, meaning it creates a double-stranded break in DNA. So it breaks both of those DNA strands. And naturally, if nothing else were to occur in the cell would no longer be able to replicate because it has a break in the chromosome. And so replication wouldn't happen properly. But fortunately, cells have this repair mechanism called non-homologous end joining. So non-homologous end joining is pretty good because it can repair that double-stranded break, but it's pretty bad because it's not very good at repairing that break. It doesn't know if there's any missing DNA in between those two pieces. And so it sort of just, in the end, it assumes what is supposed to go there. So it may delete some base pairs, it may add some base pairs. And effectively what this does is it breaks that gene. If that cut occurs within a gene, the non-homologous end joining repair will break that gene. So it effectively knocks out or deletes that gene. Technically, we don't call that a deletion. Technically, we should call that a knockdown, but effectively that gene no longer functions when non-homologous end joining repairs that break. So CRISPR does not delete genes. The CRISPR-Cas9 system just cuts DNA, and your own cellular mechanisms or a yeast mechanism or a bacterial mechanism is going to go in and actually repair that break poorly and cause that deletion, which is better than complete cell death, of course, but it's not CRISPR-Cas9 actually causing that deletion. When we do these genome-wide screens, the idea is that we have a library, again, this set of different guide RNAs that we can put into several different yeast cells. The guide RNA that we design target every single gene in the genome. So you have a single flask of yeast. You imagine that each yeast cell is going to get a different guide RNA, and that leads to each yeast cell having a different gene knocked out, right? So in a single flask, you have every possible single gene deletion. And then you can do further experiments to sort of elucidate what those gene functions are. Is that gene really essential for growth? Is it needed for a certain condition, etc.? One gene deletion may cause the cell to die, and we would call that an essential gene, or one gene deletion may improve growth, and that may be interesting for other reasons. And so we want to answer a couple of questions with these genome-wide screens. One is, what is the cutting score of the guide? So cutting score is how well a guide actually cuts. Of course, the guide isn't actually cutting. Really, this is a question of how efficient is that guide at allowing Cas9 to cut. But we call this cutting score because effectively we sort of say that the guide is cutting even though it's not actually doing the cutting. We design these guides computationally, and we may be wrong sometimes. Sometimes we assume a guide is going to cut well, but it doesn't cut well. And so we need a way of quantifying if our guides are actually cutting. And then the second question that we want to answer with these screens is, what is the fitness score of these genes? So assuming the guide does cut, what effect does that have on the cell, and how do we quantify that? So we do that with a couple of different styles of experiments. The first is I'll focus on the cutting score experiment. So we have our guide RNA library, and we put it into two different strains. Here we're focusing on Yarrowia lipolytica. So we'll put it in two different strains. The strain on the left is the control strain. It has Ku70 knocked out. If you ever see delta in front of a gene, that means that gene is knocked out. And on the right, we have our experimental strain that has Cas9 and Ku70 knocked out. So Ku70 encodes for non-homologous end joining, which allows for that repair process to happen. So if you knock out Ku70 and you make a double-stranded break, that cell should die. So if you have a good cutting guide in this strain on the right, the cell should die. On the left, you don't even have Cas9, so no cut's going to happen. And in order for it to be a good control, you also need Ku70 still knocked out. So we'll grow these two different strains in glucose media, so just regular media, and we calculate the cutting score by calculating the negative log two-fold change in the abundance of the experimental strain, which is the Cas9 delta Ku70 strain, divided by the abundance of guides in the delta Ku70 strain. So the abundance is saying, if you have a good guide, if you put that good guide in the Cas9 Ku70 strain and you grow it for a while, that guide abundance in that population is going to go down, maybe even to zero. Whereas if you have a poor cutting guide, it's going to go into that strain, not make a cut, the strain's going to live, and so you'll have a higher abundance of that guide. And so basically when you see cutting scores, a higher cutting score means it's a better cutting guide, and a lower cutting score means it's a worse cutting guide. So moving on to fitness scores. Fitness scores are calculated with an experimental strain that just has Cas9, and the control strain is just wild type. We put WT. So we use the same library here, and assuming the guide does make a cut, we have corrections for this. Assuming the guide does make a cut, the fitness score is going to be the log two-fold change of the abundance of Cas9, the abundance of the guide in the Cas9 strain divided by the abundance in the wild type strain. You imagine if a cut is made in the Cas9 strain in a gene that's essential for growth, that abundance is going to plummet. Whereas if a knockout occurs that improves growth in a particular condition, then that abundance will rise. And we have to have a control. So in both of these cases, the control population is just going to have like a normal distribution of guides, sort of a random distribution of all of the guides. And so we can do this fitness score experiment in glucose, just regular glucose media as well. But in this paper, we did it in acetate media too. So in glucose media, a low fitness score we would call an essential gene. So we would call that as an essential gene. And then in the acetate media, we might be able to pull out some high fitness score genes that actually improve growth in acetate, so that industrially, when we want to feed our yeast acetate, maybe knocking out a particular gene would improve growth, right? So we also did some fitness score experiments on solid media. So instead of growing them in liquid culture, we grew them on solid plates, and we had a slightly different calculation for finding the fitness score, but the idea was the same. And we did this in fatty acid and other hydrocarbon media, so dodecane, margaric acid, and oleic acid. So, if we get high fitness scores in any of these conditions, potentially deletion of that gene would improve growth. And sure enough, this is just showing the best growth strain that we got. But I did shake flask experiments over several hours, so this is requiring coming in at like 3 a.m. sometimes. But P01F in the blue is the control strain, it's the wild type strain, and in the pink or magenta, this is the Delta 21022G strain, which is one of the hits that we got from our acetate screen, and this is in acetate as the sole carbon source. So our best performing gene deletion halved the lag phase, which the lag phase is basically the time that it takes for a strain to adjust to a given media condition, and it statistically significantly improved the growth rate in acetate as well. So this is fantastic. In our hydrocarbon screen, these are dodecene plates that contain dodecene. Again, P01F is our wild type. It doesn't have very good growth, but in one of our top hits for dodecene, Delta A18344G, we had improved growth. So both of these figures are further validation that our screen is actually working properly. And just so you know, again, that delta means that the gene is knocked out. A refers to the chromosome. So Yarrowia has A, B, C, D, E, F, G. It has 7 chromosomes. And then the number is just the number of the gene. Now we want to implement the Pure One biosensor technology and the CRISPR genome-wide screen technology into a single system. And again, this is a project that I've been working on since the beginning of my PhD in various forms. Just as a very brief overview, we have this new genome-wide library, this guide RNA library, and instead of Yarrowia lipolytica, we're working with Cluvaromyces marxianus now to produce one of the terpenes that we have biosensor for. We're saying that through our biosensor, if we knock out a particular gene, that may improve the production of our terpene and we'll get stronger growth on our selective plates. And that will be seen as a larger colony. So then we pick one of these larger colonies and it should make more of our product. So again, brief overview, but first I want to talk about how we move this into Cluvaromyces marxianus. So, originally the Pure One biosensor platform, or the Pure One protein, was from plants. We do all of our screening in Saccharomyces cerevisiae to find new biosensors. And then here we moved from Saccharomyces cerevisiae to Cluvaromyces marxianus. And we did that with a GFP reporter, so green fluorescent protein. So the cells will glow green when the terpene that we're detecting is present. So you can see from these graphs, I can't tell you exactly what terpene it is, but on the left you have no concentration of the terpene, and on the right you have high concentration of the terpene. And so these graphs are demonstrating the sensitivity and the background of these sensors. So the sensitivity referring to when you actually get detection of the chemical, and then the background is how much response you have even when you don't add the chemical. And then on the bottom here, you're looking at cross-reactivity. So this light blue bar is the target terpene, and then the red and the yellow bars are the off-target terpene. So these are degradation products of the terpene that we're working with here. And so we don't want cross-reactivity, and we want really low background for this particular screen. And if you have questions about any of these details, you can ask them in the comments below. So we ended up going with this JUR21 biosensor because it has really low background and really low cross-reactivity. So then I designed this to be a growth-based reporting assay so that when the terpene is present, we get stronger growth. So we can look at a plate like at the beginning and pick the largest colonies, and that largest colony should be producing the most of our product. You can see here in this top row, this is how, adding the terpene or not adding the terpene, and this bottom row is adding the gene that makes the terpene or not adding that gene. This mechanism is giving antifungal resistance. So G418 is an antifungal compound, so Cluvaromyces marxianus shouldn't grow on that compound. But when we have a biosensor that drives this NEO-R reporting gene, the cell gains resistance to the antifungal compound and is able to grow. So when the terpene is present, the cell will be able to grow, basically. So in this left example, this is the control. This is no terpene being produced, so we get very little growth. In this middle case, I added the gene that produces that terpene, and so we get a little bit more growth. And then on the far right, the cell has the gene that makes that terpene, and I added a high concentration of the terpene to the media, and so we got much stronger growth. And so this is simulating saturation of the biosensor, or simulating mutation that may improve production of that terpene. Okay, so let's put it all together. So we have this guide RNA library that we put into our K. marxianus strain that has all of the machinery that we need, including the biosensor. And then we expect that there are going to be some knockouts that produce more of our terpene, and so we get stronger growth. And then we plate on G418 plates, and so that bigger colonies should produce more of our chemical. And so then we need to pick these larger colonies, sequence and find out which guide RNA was present in that cell, and then test whether or not they're actually producing more geraniol. We did this, and we found that the top two hits that we got, knocking those genes out, doubled the production of terpene. And so this is the base strain here. By knocking out these two different genes, we doubled the production of our terpene here. And you might be asking, why? Like, what's the mechanism behind the production? And as engineers, we don't typically ask that question, but sometimes we get curious. And so we did further experiments and we found that, and this is really fascinating because, again, we usually don't dig into this as engineers. Both of these strains produce twice as much of our terpene. And so I figured, a lot of times terpene production in the literature is limited by the tolerance of that microorganism to the given terpene. So usually terpenes are a little bit toxic to the cell, and that limits the production. And so we tried this. We put high concentration of terpene in this media, and we found that the control strain barely grows as we expected, and this first knockout strain barely grows as expected, and then this strain here actually grows much better. So this strain, apparently, at least we're hypothesizing that it's producing more terpene because it's overcome some sort of tolerance burden, right? And then this top strain here is producing more terpene from some completely different mechanism. So this is really cool. This one screen has yielded two completely different mechanisms for producing more of this terpene. Okay, so to conclude here, I did a functional genomic screen, CRISPR screen in Yarrowia lipolytica, found some mutations that improve growth on various low-cost carbon sources. I discovered some new biosensors for metabolic engineering specifically and moved those into Clavieromyces marxianus, and then used those biosensors and that porting over to K. marxianus to develop a biosensor-driven genome-wide screen. And then from that screen, I was able to identify specific mutations that improve production of geraniol. That's my PhD defense right there, or at least an abridged version of it. And if you're watching this right as it comes out, then I will be being hazed by professors, and hopefully I will come out of that meeting Dr. Nicholas R. Robertson. So thank you so much for watching. Thank you so much for supporting the channel as well, and coming along this journey with me. Be sure to like and subscribe to the channel. If you have any questions, leave them below. If you have any video ideas for me, leave them in the comments below as well. Thanks again for watching. Bye.